So the PowerPoint I posted for you guys doesn't have all the terms written out, so hopefully you wrote out and defined, right? At least done that much. So the basis of why we talk about genetics as it relates to bacteria, their genetics, is because these genetic changes lead to most of the antibiotic resistance that we see, right? Why is it they're resistant? Because genetically they have changed, and therefore they're resistant to the antibiotics that we use. And so we're going to look at several different ways in which they change that could lead to the resistance that we see today. And that we'll continue to see, right? Because this is a continuous process. It's never ending. So there are different processes, right? And this is just, I'm going to use these terms throughout, but sometimes it's good to just kind of have them all in one place, right? So I'm not going to read these verbatim. We're going to use them as we go through. But I'll make this available to you guys. So these are all straight from your textbook. So there are 3 different mechanisms bacteria use to adapt to changing environments. One of these is gene transfer. There's 2 different types of gene transfer, though. There's what we call vertical and horizontal, or sometimes referred to as lateral. So this is genetic information being transferred, right? Going from one organism to another. Is it going horizontally or is it going vertically? What the heck does that mean, right? So, mutation is one of the ways that we change. One of the ways that they change. And what a mutation is a change in the genetic code, right? What we call the genotype. The A's, the G's, the T's, and the C's, right? That code. The adenine, guanine, thymine. That change happens, and for bacteria, of course, they only have mommies, right? So when that change happens in mom, and that's a change that happens and it doesn't get fixed and it stays there, and she passes it on to all her babies, that's vertical transmission. Vertical transmission happens even for us as humans, right? Vertical transmission of disease can unfortunately happen for pregnant women, right? If you get a disease while you're pregnant, that can be vertically transmitted to your fetus. So if you think of it that way as well. Horizontal, on the other hand, is like mom to dad transmission. Right? Or in the case of bacteria, cousin to cousin, right? So one of my best examples of that is we believe that that's how E. coli 0157, that particular strain of E. coli, got the sugar toxin. The sugar toxin is a toxin that's also produced by another bacteria called Shigella, hence why it's called the sugar toxin. So Shigella has it, even to this day, the ability to produce this toxin. That's encoded in its DNA, right? It tells it how to make this toxin. Somehow, a strain of E. coli out there acquired this toxin. Now, obviously, right, E. coli's mama was not Shigella, right? You do understand these are 2 completely different species of bacteria, right? So we could say cousins, right? Because they're still bacteria, right? So just like we are still humans, right? So you can almost think of them as cousins to each other. So somehow, Shigella gave its cousin E. coli the genetic information on how to make this toxin. Now, which one of the several ways gene transfer happens horizontally? We don't know how it got there. But it probably happened one of these 3 ways we're going to talk about. So something like conjugation, you guys have probably heard of, right? Some other things that you probably haven't heard of, like transformation and transduction. that we're going to go into more detail after Mardi Gras. And then genetic expression of genetic information. So this one I'm going to give you a couple examples of, and then we're not going to talk about it again. But we're really going to dive into mutation. We're going to start that today and probably not finish it up until after break. And then after break, we'll go into the 3 different mechanisms I mentioned of gene transfer. Lateral gene transfer. So expression, right? So we have all this DNA in our cells, right? Do we use all of it? No. So for instance, a red blood cell. What's a very important protein a red blood cell needs to make to do its job? Carries oxygen. Hemoglobin, right? How do you know how to make hemoglobin? It's encoded in your DNA. Where'd you get your DNA from? Your parents, right? There's also a disease out there where you don't make your hemoglobin right and your red blood cells become deformed. It's called what? Sickle cell. You got defective DNA from your parents is what happened to you if you have sickle cell, right? They gave you not the right information on how to make hemoglobin. It condenses weird in your cells and it causes your red blood cells to form sickle cells. Now you can have full-blown sickle cell anemia, which means both your parents gave you crappy copies of how to make hemoglobin, which sucks, right? Or you could be what's called heterozygous. Only one of your parents gave you crappy information. So not all your cells get screwed up because one parent gave you good information, thank goodness, and you can use that sometimes, and the other one gave you crappy information. For most genetic diseases, you have to get crappy information from both parents to have that genetic defect. That's the benefit we have with having parents, right? 2 parents that contribute to the information we have. For most of the stuff that we do in our bodies, we have 2 copies. One from mom and one from dad. This is why you could be AB blood type, because guess what? One parent gave you the A antigen you make on your red blood cells. The other parent gave you the B. Now, depending on whether you need both those copies from your parents will determine on whether you end up with a genetic disease or not, right? Whether they gave you the good stuff or the bad stuff, right? How is it expressed, right? Do we need both those copies? Well, apparently, as far as hemoglobin goes, because you got to make so much of it, your cells use both copies. So it's kind of a random thing on whether the cell uses the good or bad copy if you're heterozygous. Now, of course, if you're homozygous for the defective hemoglobin, both copies you got were bad copies, all your cells, right, are sickle cells. Did I answer your question?
No. So, as far as like alopecia areata, so like, the toxic cells that grow hair,
Mm-hmm.
that would be, like if it comes from more than one parent,
Mm-hmm.
would it be a higher chance that the child will have it, or is it something that, might happen, might not, or?
Right. I don't know that particular disease and what the genetic basis of it is. So I can't answer that. We could look it up. and that's the thing with a lot of things, you can look it up. But the important thing, too, is that's why, they talk about people being carriers, where they'll have one defective trait, but they have another good one. And the good one is enough that you don't have a disease, right? Your body, that one copy that's good, your body just uses that and that's enough. But things that you make a lot of, you tend to use both copies, like the case of hemoglobin. So in the case of hemoglobin, if you don't have 2 good copies, then you're going to have some bad red blood cells and some good, depending on which copy the cell used to make the hemoglobin. Make sense? Okay. So you're said to be a carrier if you don't have a particular disease, but one of your chromosomes is defective, right? That you have it. It's personal. I'm trying not to cry. Right? It's kind of frightening. Right? When you talk about, carrying defective genes that can lead to disease like cancer. So, So it's just really scary, to say the least. So if you're a carrier, you could pass that on to your children, right? So if you have two people that are carriers, which happens a lot in Africa, right? For sickle cell, they may be heterozygous, so they don't have full-blown sickle cell anemia. They're doing okay. But when they have a kid, the copy that they gave to their kid, that each one of them had, was defective, right? They both gave their defective copy to their kid. So therefore, their kid ends up with full-blown sickle cell anemia because it got two defective copies. Does that make sense?
My daughter has sickle cell anemia. She has SS, the worst one, right? But she has like hardly any problems.
Mm-hmm.
And they tell me that it's because her body still makes fetal hemoglobin. Like, I don't understand.
Okay, so when you're very young, your liver has the ability to make hemoglobin. And so it's probably, because how old is she? She's 9. Wow. Some type of, her body is like, it's amazing what your body does sometimes under stressful conditions. So instead of that system of basically being shut down like it is for most people, because her normal system is not working properly, that one has stayed online longer. Does that make sense? That's cool. That's pretty cool. So bringing to that too, so different cells have different jobs in our bodies being multicellular organisms. So again, that hemoglobin DNA is very important for red blood cells. Where, say, a skin cell, are you making hemoglobin? No. No, it's not its job, right? Does it have the DNA inside that skin cell to make hemoglobin? It does. All your cells, for the most part, except for your sex cells, right, which are dividing up it, right, because you're only going to give half to your children, all your cells are the same. Genetically, they're all the same. But they don't all do the same thing. So it's the environment of our body that says, okay, you're going to be a red blood cell in the bone marrow, right? Those stem cells, they're like, okay, we get signals from the body, we need red blood cells. So we need you to be a red blood cell. You are going to just use the DNA to be a red blood cell. Where others are like, okay, you're going to be a white blood cell, you're going to be a skin cell, right? And so you're just going to use that information there that you need. So the environment for us is our body, right, that dictates what cell is going to be what. For a single-celled organism, a little bit different story there, right? A little bit different environment. But same principle. So E. coli, my favorite buddy, right? E. coli loves the sugar lactose. Oh my God. It's like thinks it's the best thing in the world. Does he always see lactose? No, because E. coli predominantly is in your intestines, your large intestines. When you eat things or drink things that contain lactose, like what? Milk, right? It stands for lactating sugar. It's found in milk. So anything we make out of milk, like cheese and ice cream, could have it in it, right? Most of us, in our small intestines, this gets digested, right? This enzyme breaks apart lactose. Lactose is actually 2 sugars. Glucose and galactose, maybe? I can't remember which two. But it's just 2 sugars. But they have to be broke apart before our body will absorb them. So some people, for one reason or another, don't produce that enzyme. They don't produce lactase, the enzyme needed to break that particular sugar into its 2 sugars. So instead of getting digested and absorbed in your small intestine, it travels down to your large intestine. Guess who's in the large intestine? E. coli. And he's like, oh, lactose, yay! Right, but he doesn't always see lactose, right? So does he have the enzyme to digest lactose? No, but does he have the ability to make it? You betcha, right? So when the environment changes, right, lactose comes along, he's like, woohoo, lactose, I'm going to make my lactase. I'm going to be eating some sugar, baby, right? And he's got in his DNA how to make it. So he's going to make it. And he's even going to make more proteins in his membrane to take in all that lactose. So if you've got all that energy, right, what are you going to do? you're going to grow. You're going to make babies, right? So all of a sudden you have E. coli, right? One cell becomes two cells. If you started out with 10,000, 20 minutes to a half an hour later, y'all, that's how fast this guy can double under ideal conditions. So we got 10,000 E. coli. Half an hour later, say, you've got 20,000. Then what? 40,000. Then 80,000. Are we meant to have that many E. coli in our intestines? No. We're not supposed to feed those guys like that. It's bad news, right? So people deal with lactose intolerance. So they may get kind of bound up where they can't go to the bathroom, or the opposite problem, right? They're running to the bathroom to get that stuff out of the system. Too much going on. Once you figure it out, you pretty much stop eating lactose, or you take lactase enzyme whenever you eat lactose, so you don't have that problem. You don't overfeed your E. coli. Does it make sense, though, how you've got that information, right? You've got that DNA, but you're only going to use it when you need it. Because it would be a waste of energy to have made that enzyme if you don't ever see that sugar. Which for most of us, our E. coli in our gut never sees that sugar. We eat it first. We absorb it and use it first. Make sense? All right. So this is my last thing, and then I'll let you go. So vertical, right? So here's a little picture for us. Mommy spontaneously mutates, right? She's copying her DNA. She puts the wrong nucleotides there, right? The code is wrong in this section. She passes this on to all her babies. They've all what we would call have become mutated. They're different from the original form. Here's an example of horizontal. And one of the common genetic pieces that will move in horizontal is actually a plasmid, right? So not even some from the chromosome, but one of those circular pieces of extra DNA that might contain antibiotic resistance on it, right? Or how to make that damn Shiga toxin. I don't know if that's chromosomal or plasmid, though. So this goes, right, from one cell to another, cousin to cousin. And there are 3 different mechanisms which will get it there, and we'll talk about those after break. But once he has it, or she has it, that then gets passed on vertically, right? So stuff moves horizontally, but is going to continue to be passed down vertically. Make sense? And so the same is true for vertical. Notice this mutation that happened here could then maybe later be horizontally transferred to some other cousin in the community. Make sense? So I didn't didact most of this. I left out definitions in a few places. Just make sure, try to read over. And again, as I said to you guys in the past, focus on the objectives, which I go over, right? Reading in your textbook, right, beforehand, right? I wanted you guys to read before coming to class, right? Try and fill in these definitions. Try and read about these processes in your book, and then I'll tell you my cute little stories, right, to put it all together. All right. Please come back to me safe after Mardi Gras in one piece.